Wonderful. Well, thank you all for having me back. It's always a pleasure getting to spend time with you all, even if it is just virtual. Yeah, and for those of you who maybe don't know who I am, I'm Dr. Nick Famoso. I'm the Paleontology Program Manager and Museum Curator at John Day Fossil Beds National Monument. But my origins are, very different from just that. I wasn't here all the time. So I grew up in Southern Oregon. I'm from Grants Pass originally, so I grew up down there, well before NARG was a thing. Which, man, if y'all would have been around when I was a kid, my mom would have been driving me up to Portland all the time. That certainly did not happen because you weren't around. So yeah, so I grew up in Southern Oregon, went and got my bachelor's degree at the South Dakota School of Mines, and actually, I started at School of Mines right the same summer that the Mitchell monster skull was found, right? So that was the same summer. So I'm moving to South Dakota while also working for OMSI as a science camp counselor, right? And I'm hearing as I'm working with the paleo research team about this cool stuff that NARG found that's going to South Dakota School of Mines, and I was going there at the same time. So that was really cool for me. Um, Ended up doing a couple internships, like the one at Ashfall Fossil Beds, like I mentioned before, and then moved over to University of Oregon to do my master's and my PhD. And while I was at U of O, finishing up in the last year of my PhD, I got hired out at John Day Fossil Beds. And I've been, working there ever since. I had a few other stints working for like Badlands National Park, the University of Nebraska State Museum for a couple of years there doing highway salvage work all around the state of Nebraska. And now I'm out here in Oregon, and I didn't think about this, but I should have had some things to share with all of you for show and tell because we've been doing field work this week. We've had some researchers from the Western University of Health Sciences in Lebanon, Oregon, out, and they've been wanting to find some of our fossil primate material, the Echemoschachalum material. So we've been targeting specific sites looking for similar material. And yesterday was a fun day because we went to a site that had a lot of rodents. So we pulled out 4 or 5 different taxa of small rodents or rodent-like animals from the horizon that we were working at, and that included a couple of mountain beavers, a chrysitid, so a little mousy mouse, some rabbit material and whatnot, but no primates yet. I didn't go out with them today, but we'll also be going out tomorrow. But it might have been hard to show you those mouse teeth on my webcam here on my computer, but it's pretty cool stuff. But at any rate, all that aside, I'm excited to talk to y'all about one of our cool things that I've recently published on. So I'm a vertebrate paleontologist, and now I have published a paper, co-authored a paper describing trace fossils and insects, and that's something that I never thought that I would ever publish on. But this was a really cool project, and I'm excited to get to share a little bit more about this with y'all. So just kind of a little bit of background to start with. So fossil insects in North America, they're not very well distributed, right? There's not a lot of distance here. This is a screen grab from the paleobiology database, which is occurrences of published records of fossils, and this one is just Cenozoic insects. And yeah, there's quite a lot of material from all over the place, but like if I put vertebrata up here, it would be absolutely chock full across North America, right? So and I've already had this paper is already in there, so there is a little yellow dot right where the fossil beds are, my part of the fossil beds where these fossils were found. But in general, most of these occurrences aren't traces or eggs. They're typically body fossils, so they look more like this, right? So these are 2 specimens out of the Clarno Formation. They're from White Cliffs and Horse Heaven Mine, which is not within the monument. I believe both of these sites are private. One of these might be BLM. I can't remember for sure. But this is typically what we find when we think of insect fossils, right? Lake bed deposits typically with complete bodies preserved. There's a cicada on the left and a water beetle on the right. And this particular one of those is from University of Florida. Actually, they're both from University of Florida on loan to us. So, thank you, Steve, and everyone down there at University of Florida for letting us have these on loan. But that's not the whole story, right? And this is typically why paleobotanists tend to get really tied into insects simply because the insect fossils tend to be fossilized in a lot of the same deposits that the plant fossils are, usually those lake bed deposits. And these fossils are great, but we don't have a ton of lake beds, especially in our well-known Turtle Cove assemblage. And what we have instead is a lot of overbank deposits, flood deposits that preserve other types of fossils. And what we typically find here are trace fossils, so things more like this. So we've got sweat bee cases, dung beetle balls, earthworm mounds, termite mounds. These are things that are more commonly found in our deposits here in the fossil beds. And all of these are actually from the sheeprock unit from the Turtle Cove members specifically. I'm not aware of a lot of other structures from some of the other formations that we have, but I believe that there are some other structures from the Maskell Formation. But definitely, this is the key that we have here in the Turtle Cove in the Sheeprock unit, which is pretty cool. I never really thought about traces before, but we have quite a lot of them. And those dung beetle balls and sweat bee cases are everywhere when we're looking around for fossils for the vertebrates. But so specifically, we found these interesting fossils that we have shown in our paper are grasshoppers, right? And just to kind of give you the distribution of grasshoppers in grasshopper fossils in North America, it is very sparse, right? We are the only site that has anything of grasshoppers in the entirety of the Pacific Northwest. And there's only four sites if you count Canada, west of the Rocky Mountains, right? And I didn't look to see what kind of fossils they were, but I know for a fact that they weren't eggs or egg pods because these ours are the first in the world that have ever been described and confirmed to belong to orthopterans or grasshoppers and their relatives. But just wanted to give you a distribution and an idea of the fossil record of these things, which is not super big. So the collecting history. So for the longest time, what we would find are these isolated eggs or small groupings of eggs, like what are seen here in the figure there. They look typically, not the bottom three are kind of making it seem like they're not all white, but most of the time they're white elliptical fossils that are about the size of a grain of rice or a tic tac. Those are often the jokes I would make when we'd find them out in the fields, like, oh, we found another tic tac, right? Because that tends to be what they look like. Originally, in our database, at least, they were thought to be ant pupae or ant eggs. Sometimes there would be comments about them being bees or wasps, something like that. And in fact, these were labeled as ant eggs in our visitor center exhibits that opened in 2004. Well, 2005 is when it was formally grand opening, but the visitor center technically opened a soft opening in 2004. But we were calling them ant eggs, right? And the very first of these were collected in 1996. So we've had them around for a while, but not a whole lot of work was ever done to really try and figure out what they were. And when we find these small clusters, there's only so much that you can glean from these small clusters. Saying, oh, well, that's cool, but you can't really tell, like, were they in wood? Were they in something else? So it took, finding something a little bit more complete. And so back in 2012, my collections manager, Chris Sharp, was out on this outcrop and was able to find a interesting fossil. It was about the size of a fist, roughly, maybe a little bit bigger than that, that included a cluster of these fossils. So, this was in 2012, and they set it aside in the accessions, and it didn't get cataloged, I should say, until I got here in 2016, and he showed it to me, and I said, that seems pretty important. We should probably catalog this. And then I didn't really look at it again until probably like 2018, 2019. Oh, it was 2019 because there was a paper that came out that started describing insect eggs, because turns out no one ever thought to describe insect eggs of the modern variety at all. So I started looking at that and started making some determinations on that. So it is kind of a long history leading to where we are now, and I'll get to that a little bit later. But so this is the area where the nest was found, is what it looked like. It's a very steep slope, and we're lucky that, it probably only tumbled a short distance down from the top of the hill there. But we have some other known localities. So, right, if you don't know where we are, right, here's our map of Oregon, and all of these fossils were found in the sheeprock unit, which is the small purpley dots there, kind of on the far right. Painted Hills and Clarno are the ones on the left. Um, but all the fossils that we had in the paper were from the sheeprock unit. And the fossils in the stratigraphy are all laid out there on the right-hand side of the stratigraphy. Each of those are little grasshoppers silhouettes in there, and those all represent horizons where we had confirmed grasshopper eggs from. And the names of the localities up to, or the locality numbers are on there, and these are from there's a site kind of north or south of Blue Basin where we got a couple of those. Some of these are from Blue Basin, and some of these are from 4E. The type is actually from JDNM 9, which is Blue Basin. That's right. That's where that one's from. So that's why it's in red, is to indicate that that's like the type specimen that we had for these specimens. Um, and yeah, so they're fairly limited on where they are, but they're distributed pretty broadly across all the different horizons. So if you're not familiar with the Turtle Cove member of the John Day Formation, it's kind of an alphabet soup of different horizons going from A to K2, and most of these are separated based on ash layers, many of which are dated a lot more than what we have there. We have another paper that'll be coming out hopefully later this year that will increase the number of radiometrically dated ashes, probably double or triple what we have in here. So there's a lot of new dates that we have that are very high accuracy, about plus or minus 10,000 years. So that's really cool that we have that. And most of these are smaller ashes, but that picture gorge ignimbrite in the middle is what I like to call fiery cloud of death. It's about 100 feet thick deposit that would have been a superheated gas cloud coming out of a caldera in Prineville, Oregon, what's now Prineville, Oregon, and rushed over to this area, wiping out a lot of the habitat that was here at the time, which was forested for most of that period of time before that ignimbrite. Afterwards, it seems that it was more open, and that's interesting because we don't seem to find as many grasshoppers at that particular time, at least occurrences of them from the eggs. They seem to be very common in the lower Turtle Cove. But yeah, so that's a pretty interesting, thing that we're seeing there with all of that, and they're pretty localized with where we've found them before, and that could be preservational or it could be environmental. It's hard to tell at this point with as little as we know about them. We also, so at the time of publication, we only knew them from National Park Service sites within the sheeprock unit of the park. I went through the database a couple of weeks ago and did discover that there is one egg that is described as a termite that I missed because I wasn't looking for termites. Don't know for sure if it is in fact a termite or one of these grasshopper eggs. It might be something different, but it's from much higher up in the section, from K1 or K2. There is also one specimen that I didn't notice before either that was from a Bureau of Land Management site that's up by the Clarno unit. And that site is Lower Turtle Cove, so that seems to fit with where we find most of the majority of the other fossils, so that's not too surprising. And then I was informed recently that there may also be another nest that's in the University of Oregon collections. I haven't seen a photograph of it. The lead author on the paper, Jabin Lee, was the one who told me that someone from U of O reached out to him about the specimen. So there may be a few more that we didn't know about when we publish the paper. But that's great, right? That's kind of what we hope and expect, right? Now the paper's out there. I even got someone from my old advisor from South Dakota School of Mines was out doing field work and sent me a photograph of something that looks very similar to this in South Dakota. and said, "Is this like what you have?" And I said, "It looks a lot like it, but they're like way smaller. So maybe not this type of grasshopper, but maybe some other insect egg," which is pretty cool. And so now I'll get to show you the photographs of what the nest looks like, right? So this is the nest that Chris found in 2012. Scale bar on the bottom there is in centimeters. So, right, I mean, these are not very big. And what's interesting is that they're in this radial pattern. And it does look like they are stacked. It looks a little bit better in this version of the photograph. You can kind of get some depth perception there, that there is some stacking that's going on with these eggs, right? And they're very shiny. And I will say that there was no preparation on this specimen whatsoever. This is basically unmodified from the way that we brought it into the field, other than a couple of pieces of rock have fallen off over time from the base, not from around where the eggs are themselves. This is a very delicate specimen, and part of the reason why I made sure to put it into our type cabinet, because I didn't want anyone touching it, or I wanted very minimal agitation on this specimen because it is very, very delicate, right? So this is the primary specimen that we had that I got micro CT scanned. I also got A and B CT scanned as well, because I wasn't sure how high resolution we'd be able to get with the nest, but I wanted to look inside of the eggs as best as I could. So I had a couple of isolated eggs, and I also had a couple of small clusters. Now we also found this other interesting cluster that's in C, that's a little bit more mishmashed there, and it may be that this one got damaged through some sort of taphonomic process, but we don't know. We didn't get that one CT scan, but it's pretty cool nonetheless. So as far as the CT scanning goes, so I'm a courtesy faculty member at University of Oregon, and so I chose to use the micro CT facility at the Knight campus down at U of O to get the CT scanning done. So that was done with my other co-author, Angela Lynn, who's the lead CT person at the Knight lab down there, and she took these specimens and stuck them into her fancy micro CT machine, and ran all night long. Basically, it's computerized, so it'll like pick up specimens, put it in front of the CT scanner, scan it, and when it's done, take it out, grab the next one, put it in. So we were able to run that overnight, so that way I didn't have to be there for a whole week. I was only there for like 2 days. And you can see some of the statistics or some of the data on the different CT scans there. And the insect group, the small grouping of eggs, only took about 2 hours to run, but the nest and the 3 isolated eggs took 5 to 6 hours to scan a piece. And also the important thing I wanted to point out there is the microns there. So the voxel size, which is the distance between each of the scans, is between 7 and 30 microns, which is very, very small. So all of these slices are incredibly tiny in order to get this high resolution on all the specimens. And what did that look like? Well, it's not as easy to see in this, but the one on the left, those are the 3 isolated eggs. And turns out they're hollow mostly on the inside. The other thing that's maybe a little hard to see on the projection that you might have there, but there's little fingers coming down off of the edge. So this is essentially filling in like a geode does, right? There's a hard rind on the outside and crystalline growth that comes towards the center of this object. And this is another reason why we knew this wasn't some modern thing that got deposited, that this had to have been fossilized just because of the level of mineralization that has occurred. In EU, we can see that some of these are partially filled. The lead author, Jae-min, thinks that some of these may actually have some embryo preservation. We need to do a little bit more looking into that to see if that's actually the case. But definitely some of these are hollow, some of these are partially filled. It's really interesting. And from the CT models or the CT data that we had, we were able to create 3-dimensional models in high enough resolution that we got the equivalent of an SEM scan or a scanning electron microscope scan of the surface of the eggs because that turns out to be an important texture that you need to use to describe insect eggs. So that's what's in part F of this particular figure, is that texture on the surface. And so what the nest itself looks like, right, from this we were able to determine that there were actually approximately 50 eggs in this, which is a pretty large clutch. And it may be a little hard to see here. I'll try with my mouse if you can see that. But there's this light white ring that we're seeing around where all the eggs are in nearly every single one of these scans. It shows up really well in the bottom row C there. What we're interpreting that as is as a protein slurry, which typically gets deposited when subterranean grasshoppers lay their eggs. So and I have some pictures of what that looks like later, so you get a better sense of that. But there is like some goop, I guess, if you want to call it that, that comes out of the grasshopper as it's laying the egg, creating the egg pod. And that's what we're interpreting that lighter color to represent, right? So clearly it's a different density from everything else. And so that's really cool that we're able to see something like that in this particular case. And then my co-author, Jay Min, spent some time trying to segment out from the CT scans the eggs from the rest of the pod, so the sediment around it, and that's what this figure here represents. The yellow are the eggs, and the bluish color is the nest itself, like the egg pod itself. And so you can really get a sense of the way that this was arranged, the way that it was deposited. So we see a semicircular radial pattern to this, and just kind of that interesting way that it is getting deposited, like in part F there. You can really see that those eggs are all kind of in a, almost a curve with the way that they were getting deposited. And if you are interested, the CT scans and the model are available on Morphosource, which is a digital repository for three-dimensional data. The links up there. There's also DOI links that are in the paper, which is also an open access paper. So if you all want to, see what these look like, or even play with the data, spin around the model, you'd be able to take a look at all those things yourself. But just want to describe a little bit here about the specific characteristics about this nest. So it's largely, it's made up of large elliptically curved eggs. And those eggs, according to that 2019 paper on modern insects, fits really well with the description, both the ratio of length versus width, the curvature, all of those sorts of things fits almost perfectly with grasshoppers, right? And the size also fits better with grasshoppers than especially ants. I started with ants and I was like, wow, these things are like 3 times the size of what we would expect ant eggs to look like, even large ants. So it's like, this doesn't seem to work, right? The other thing that's interesting about this is it's a fairly large clutch size, so 50 plus eggs that are in there. That's quite a lot of eggs. And like I mentioned before, the radial pattern is important, and that protein rind is really important. So if you've never seen subterranean grasshopper eggs or grasshopper nests, this is kind of what that looks like. A grasshopper sticks part of its body down underneath the soil and sort of lays these eggs in a cluster. You can kind of see in the far right, there's this kind of white silky looking stuff. That's the egg pod itself, so the material that the grasshopper was using from its own body to sort of create this more moist clutch area there. So that's what it looks like for modern. We are working on some art with an artist named Reed Saltis, who was from Portland and now is down in the Southern California area. This is a piece of draft art that he's putting together for us to kind of get a sense of what exactly that looks like when these grasshoppers lay their eggs. This is not the final product, but there will be a much better one. But this sort of tells the story about what we think these things were. This is modeled after an Indian rice grasshopper, which lays its eggs next to bodies of water like rice paddies. And that's what we're interpreting this as being laid next to fairly wet environments. So some of the cool things that we were able to do, right? We can't describe the grasshopper because we don't have a body fossil, but we can describe the nest and the eggs. And so describing the nest, which is technically a trace fossil, right? So that uses ichnotaxonomy, which is basically which is a way of communicating information about these structures, which is best done by naming, right? So, there's all kinds of trace fossils. Usually we think about ichnotaxonomy for trackways, but you can also do this for many other types of traces, including the dung beetle balls, the sweat bee cases, all those other things. They can all be classified using an ichnotaxonomy. And we ended up naming an ichno family as well as a new ichno genus and a new ichno species. And then we also, for the first time, utilized something called ootaxonomy, which is using the same sort of naming conventions like we do for family, genus, and species for other things, for plants and animals, but we also use that for eggs. Typically, this is done with amniote eggs. So like, chicken or bird, I should say, dinosaurs. That's typically where people have focused is birds and dinosaurs, but this also has applications for insects. When you find them, I mean, they're rare, but we do find them, right? And as I mentioned before, this is the first ever confirmed grasshopper egg pod and grasshopper eggs in the world, right? There's never been fossilized ones of these confirmed any other place in the whole entire world, right? Which is pretty cool. So the names that we decided to go with, I left out the family names here, but these are the more important ones. So the genus is for the ichno species. So for the nest, it translate. I'm not going to try and say them, but I will translate them. So the genus names translates for the first one as underground egg pod trace fossil, and the species name for this is radial, right, which describes the structure of what we have. And then the Uo species, the genus name is curved elliptical insect egg rock, essentially is what that translates to, which is a great way of describing it. And we couldn't come up with a good way of describing a good name for the species, so I opted to name the species of these eggs after the first superintendent of John Day Fossil Beds National Monument, who was Benjamin Ladd, who unfortunately passed away a few years ago. So this was posthumously named after him. Partially the reason why I did that was because all these fossils were from National Park Service land, and Ben was the superintendent who really set up the paleontology program, hired the first paleontologist, made it a priority to protect all these resources in and around the area. And without that groundwork that he laid, there would have been no possibility of really finding something so delicate in the way that we were able to find it. So in conclusion, right, these eggs and large egg clutch and things are known from no other living group other than grasshoppers and locusts, right? So that's how we know for a fact that this has to be from grasshopper locust family. This is the first ever grasshopper egg and egg pod recognized in the world. We're formalizing the description of these immature stages of insects using the Uo taxonomy, which again is really hard to do anything other than adults. It's very difficult to find in the fossil record. And particularly because these are all overbank deposits, right? It's like incredibly amazing that we were able to preserve these things in that sort of environment in the first place, because it's a lot higher energy than those lake bed deposits. So that's really exciting. And this gives us additional data on the evolution, behavior, and ecology of insects in deep time. And it gives us clues into the environment where they were fossilized, since they most likely were deposited in a wetter sort of environment. So then I've got these acknowledgments here, my co-authors, Jaemin and Angela. Part of the reason why Jaemin was able to get involved with this project was because there was a field trip from UC Berkeley led by Jack Sang and Cindy Louie, who came out to the park, and I then found out that he maybe had some insect background, and I looped him into the project through that. Chris and Jennifer, my two employees, were important in getting figures made, photographs made, and obviously Chris found the thing in the first place. And this was published in the journal Park Stewardship Forum, which is an open access journal focused on national parks. And we had a special issue that was paleontology based, that was refereed by Vince Santucci and Justin Tweet from the national office. I did also want to just mention real quick and acknowledge that the fossils that we found here are from traditional territories of the peoples of the Confederated Tribes of the Warm Springs and the Burned Paiute Tribe, as well as traditional territories of the peoples of the Umatilla Indian Reservation. And with that, I can take any questions if we have any time. Thank you.
Thank you, Nick. Yes, we have time, and yes, we have questions. So
Okay.
here, bear with me for a second. Make sure when you're asking the question, you state your name.
Fine. Hi, I'm Elena Cancelon, and I wanted to know, how did you come up with the names? Like, do you know, was it Latin?
It was a combination of Latin and Greek that were used obviously for the genus and for most of the genus and species names, and we did Latinize Ben's last name for the Lati part of the Uo species. But yeah, it's typically, I think most of that was Greek and a little bit of Latin where it was appropriate. Yeah.
Okay. Do you had to study and understand, or did you just Google it?
So J-Min is the one who spent most of the time looking that up. But I believe, like, there's Latin dictionaries and Greek dictionaries and things like that. So that was probably a lot of like what we were looking up and finding, like, what was the word for subterranean, right? It's subpterouthekia, right? And euthekia is like a word that we use to describe egg pods in general. So that's why that was part of the root of that first word of the ichno genus. And ichnus is also a really common phrase used to describe ichnofossils. Um, right, so that was a lot of where that was coming from, was like using that Greek and Latin. I don't know if he Googled it or if he used a dictionary. Some of those words are just common for the language that he knows as being a pseudo paleoentomologist, but yeah, so it's probably a combination of all three of those things.
Alrighty, thank you. That's all.
Okay. I did have to Google the appropriate way to Latinize an English name. I wanted to make sure that it was just simply putting one I on the end of it, because sometimes Latin is weird. It's different when it's male versus female. It's a whole thing. So I did have to Google those particular rules to make sure I did it right. But yes, thank you.
I get it.
Yeah, great. Good question, Elena. Anybody else have questions for Nick? Yep, okay. Lucy's got a question, heading back to one of our guests.
Yes.
All right.
Hello.
Hello.
So could you tell me what autotaxonomy was again?
So the Uo taxonomy, that is the naming of genus or genera and species of eggs, right? So it's using right, it's like form taxonomy, like describing a form of things, but specific to eggs.
Mm-hmm. And what's icrotaxonomy again?
Yeah, so ichnotaxonomy is utilizing that same sort of structure, but to describe trace fossils.
Thank you.
You're welcome.
Great questions, Lucy. All right. Yep, got another one.
Hi, so my question was kind of related to like the Uo taxonomy thing. I know in some branches of paleontology, it's common to like have separate taxonomy for different like parts of things, like in paleobotany.
Yeah.
I didn't know that was a thing in like other parts of paleontology. So is that fairly common for when they find like fossilized eggs or things like that?
So Uo taxonomy is not very commonly utilized in general. And like I said, it's mostly focused on like eggs of amniotes. So I think it's mostly used, has been used for like birds and dinosaurs. But the literature on it is not very in depth. There's like a paper in I think the 90s that was like, this is something that we need to do, right? Because I mean, how are you really going to be able to attach the eggs for sure to the animal that laid them unless it dies with the egg inside of it, right? Like it doesn't make a lot of sense. And it's also possible, just like with form genera, form species, form taxa in plants, that you can have like a similar egg between multiple species of animal So it's just another way of just describing like this is right. It doesn't mean the same thing as an animal genus and species, right? It's another way of describing that using a similar frame. But yeah, it's not very commonly used. The ichnotaxa is much more commonly used. And I will say there is a formalized body that sets rules for the naming of animals, and another one that sets the naming for plants, right? So the ICZN and the ICBN are the two that set the rules for what that looks like. But with ichnotaxonomy and ootaxonomy, it's a wild west. There's no rules. So who knows? In the future, someone may have to restructure all of these names that we did if that's ever formalized. But yeah, at this point, there's not a lot on ootaxonomy just because it's new and people haven't really thought about it, mostly because these aren't very common to find.
Gotcha, thank you. Are there other kinds of like form taxa in vertebrate paleontology that are similar to that, or is it kind of just those ones?
I think it's typically when you're working with vertebrates, it's usually just ichnotaxa. And like I'm working on another project with a student from Utah who's describing all of our vertebrate trackways. And that's like obviously we can only use the ichnotaxonomy for that. But so I think ichnotaxonomy is the most common sort of form thing. But oftentimes what I've seen too is when we don't have enough of an animal, will oftentimes describe morphs very similar to what you do with plants. I mean, they're not formalized morphs or anything like that, like what you get in paleobotany, but like sometimes I've seen like, well, this is like this genus species A, this species genus B, or this genus species B, because we don't have enough data to really be to know for sure what it is. But we know that there's different morphologies, but there's not enough there to describe a new species. So that's yeah. So sometimes we do that, but most of the time when you see those sort of form things, it's going to be ichnology.
Gotcha, thank you.
Welcome.
Yeah, we got more. So, all right, Bernie.
My name is Bernie, and the presentation's been amazing, but I was wowed from the very beginning when you said that somebody found this in the field and somehow got it back to the lab, crumbly and falling. It could have fallen apart at any point, and apparently was not wrapped in a plaster jacket or anything in order to transport it. Can you describe? What did what did they actually do to get this thing back, and understanding how important it might be?
Yeah. So the specimen was wrapped in toilet paper to try and like keep it from, losing too much like off the side or getting brushed and damaged in that way. And he hand carried it back. So where did I say that was from? That was from Blue Basin. So Blue Basin, there's a couple of trails that go right through the middle of those areas, and so it was easy to access the site for the field crew that particular year and get back to one of the trails. I don't remember where specifically that was in there. And hike it back in, and from there it's maybe like a mile and a half to get to the parking area where the truck was left, and then drove it back to the visitor center and brought it into the lab, into our accessions area there, right? And also just noting too that Blue Basin is closed to off-trail traffic in general because there's so many delicate things like this in that area, and we don't want the general people, not obviously like people like you all are a lot better at identifying important things out in the field, but most of you are not, or you all are not the majority of who comes out to our park, right? Most of the time we get 200,000 people a year. So that's why we have that closure. So the closure is important because of how delicate things like this is, and this just kind of underscores that importance of having, people who know what they're doing collecting things and bringing them out. Because I mean, I was freaked out when I was driving this across the Cascades to Eugene. You should have seen the way I packed this thing. You would have thought I had a bomb in there because I was so worried about it going off, right? But it was just super packed, lots of fluff, lots of stuff. I was even worried about the fluffy stuff ripping it apart, so I put Tyvek around it, right? And that's how I transported it over there. I had it suspended in things. seat belted in, right? I was making sure that nothing happened to that thing, and luckily I made it. It made it there and back without any major disasters. So yeah,
5
it
miles an
was
hour the whole
yeah,
way.
it was hard.
Great question. Thanks. Elena's got another one. Yes.
Hello, it's
Hmm.
me again. I was wondering, have you identified any embryos and any like egg sacs that are still inside there, either crystallized or fossilized?
Yeah, so my lead author, J-Man, when he was looking at some of those isolated eggs, particularly the ones that are partially filled, he was thinking that there might be some sort of embryo preserved in them, but almost all of them are hollow. So we haven't seen anything really in there. What I'm imagining probably happened was that, right, the eggs were laid underground. There was another flood that buried them, created an anoxic environment. Eggs died and rotted away on the inside, but left these hollow voids behind. And then the water was able to percolate through and leave different minerals in the void to create the little geodes that became what we're now identifying as the eggs. Um, so not a whole lot of possibility for that, but we need to look a little bit more closely on the CT scans to see if we are seeing any new anything that could be an embryo. So nothing yet, but maybe more later.
All right, thank you. That's all.
Thank you.
Lori Cancelon, I wanted to ask about the dung beetle balls. You said there are tons
Yeah.
of those things, right?
Mhm.
So has anybody cracked those open to see what the content is in those? Because wouldn't
I don't know if
think would be
anybody
in
has.
them?
Right. I mean, like we have had coprolites and things that have come out before that have like little pieces of bone and stuff like that. I don't think that anyone has done anything like that. If anyone has, it might have been Greg Rotalick from formerly of U of O. But I am not aware of anybody who has done something like that to see what's on the inside of them.
Robert would say, would you ever,
Oh, I'm sorry, it's cutting out a little bit. I couldn't hear you.
would you ever put preservative on that specimen?
I don't we talked about that and we were too worried about applying consolidant to that just because we were worried that it would turn it to mush and then we'd lose all of the structure itself, the important structures of the egg. So we're really hesitant on doing something like that with this particular specimen, just because we're concerned about how stable it would be if it got wet in that particular way. So for at this time, we're not going to do that, and it's probably good that we didn't because sometimes those consolidant glues can modify what you see when you do a CT scan. So it was really good that we didn't do that, so there was no question that what we were seeing were actually like natural structures and not something added by by preparation work or stabilization in that way.
Great. Thanks. Other questions in here? Greg, you go.
This is Greg Carr. What journal was that published in?
Hey, Greg. Yeah, so it was published in the journal, oh gosh, Park Stewardship Forum. It's a publication by the University of California at Berkeley.
How about online? Is there anybody who wants to jump in online with a
Looks
question?
like we've got one hand up.
In
Yes,
online.
Dr. Famoso, how do you identify, but more importantly, verify that your dirt clot / concretion thing is an insect trace fossil? Because I know, like for me, if I was in the field, I would probably doubt myself that this round thing is in fact like a dung beetle ball or what. So what do you look for when you're making
Right.
that determination?
Yeah, so the dung beetle balls tend to have like they're round, right? And they tend to have a little pinch on one side. And you know, if you just found one of them, you'd say, "Oh, well, who knows what this is?" But like we've found like hundreds of them and they all look the same, right? And they have been published on before. Like I believe Greg Ritalik or some of his students have published and said this is what we think it is. So it's oftentimes just looking at the publications and confirming, like, okay, other people who know way more about these things than I do have interpreted them this way, and they look like the same thing with the sweat bee cases. They look a lot like the brood chambers of other sweat bee cases, or sorry, they look like the brood chambers of sweat bees that have been described in other places, like in Badlands of South Dakota. That's actually where a lot of these structures are being compared to. Like the dung beetle balls and the sweat bee cases are also seen in the deposits at Badlands, which also have fossil soil deposits, like what we have here in the John Day. So it's mostly just comparing to what we have in those other places. Because I'll tell you, vertebrate guy, I don't know a whole lot about anything when it comes to these things, and so that's why it was really important to later bring in somebody who could actually had, you know, knows a little bit more than I do to confirm that my hypothesis was correct based on all this body of literature. And I will say that the paper that describes these fossils is about 70 pages long, and it's because Jae-min wanted to make sure that there was no doubt in anyone's mind that this was what we said it was.
Wow.
Right. So it's just a lot of confirming with other pre-existing literature, finding the right number of specimens to say, like, okay, yeah, we have a pretty large sample size to confirm that this is legitimately what it is, and then putting it into peer review to confirm that that's what it is. So that's kind of how this all went down, to confirm that that was in fact what we had.
So
Great.
those are some easier shapes to identify, like the sphere and the oval look a little more natural, like when just seeing them. How do you identify some of the more like complex shape stuff, like coprolites, which could, you know, not always look like a nice coprolite,
Yeah,
look a
so
little more
coprolites
deformed?
usually, so herbivore coprolites are a lot harder to identify. Vertebrate or carnivore coprolites are a lot easier to identify because there's usually big chunks of bone that are very clearly crunched and in a shape that looks like a coprolite, right? Like scat. Same thing with, I've actually also found owl pellets or some other kind of raptor pellet before, fossilized ones. And that one you can definitely tell because it looks like that. There's a site that I go to where I actually have a fossil hedgehog skull that I'm working on discovering or working on describing. And all the skulls from there have the backs of the skulls crunched, gone, missing. And they're mostly gophers with the occasional thing like a hedgehog. And that is characteristic behavior of birds of prey capturing rodents and crunching them, right? So like you just like oftentimes we have to look at other sort of modern systems to get a sense of, okay, well, what looks like this, right? And like with the coprolites, it's just a matter of, okay, what does the shape look like? And like, oh, there's a bunch of broken up bones in there. Okay, that makes sense. This is probably a coprolite. And for whatever reason, the coprolites do stick out a bit. It is actually interesting over at Ashfall in Nebraska. We had a couple of things that we were identifying as coprolites, but also a few things that were being described as vomit, fossilized vomit. And there's like reasons why it was described that way. And there's a name for that. I just don't remember. It's been like 15 years since I talked about any of that stuff, so I've forgotten most of it. But you know, so it's just a matter of comparing it to modern things. And oftentimes these structures will be supported by you know, proteins or other things that are in whatever excreted it into the environment, and that helps to sort of keep it together and fossilize a little bit differently than the rest of the rock that's around it.
Thank you.
You're welcome.
Great. Other questions from folks online? All right. Thank you so much, Nick, for taking
Yeah.
the time.
You're welcome.
See and hear the clapping. And if you could send, oh, there's Mackenzie's clapping. See that? If you could send back the host to me.
Oh, yes.
And before you leave, you're welcome to stick around and listen to the rest of our meeting, which is a business and eating cake. Sorry you can't be here. I guess we could mail you a piece of cake.
Oh, that's okay.
Okay.
I had a piece of pie earlier at the cafe in Dayville, so I'll take that as my pie,
Oh.
as my cake.
Yeah, and some of us know quite well about the place you get pie in Dayville. That's yeah, if we still talk about that place, that's so fantastic.
Yes.
If you aren't too busy in first Saturday of August, we invite you to join us for our Fossil Fest. So we'll be celebrating.
If I can make it out, I would love
20
to
years.
come out and visit.
We'll remind you. All right, thanks.
Okay. Thank you.